Dementia Researcher with a blog and a rating.
Hello, my name is Ajantha Abey. I am a PhD student at the University of Oxford, and today I'm going to be reading my blog on the use of AI for fundamental research, part 3 of my blog series on the use of AI for dementia research. What are the potential applications, benefits, and risks of AI for dementia researchers, patients, and research itself? What is the most optimistic scenario for how things could turn out if we employ AI in dementia research, and how do we work towards this end while mitigating any potential harms? These are the questions I've been trying to address in a series of now 3 blogs. In the first blog, I discussed how individuals can use AI in personal, day-to-day research tasks. In the second, inspired by a debate at the 2024 AI UK conference, I discussed the opportunities for using AI in dementia diagnosis, trials, and healthcare, as well as the potential harms. The question of the debate was actually much wider though, asking what benefits AI could bring to dementia research as a whole. So in this blog, I wanted to discuss everything else. How can AI assist us at the basic science level of dementia research? You might imagine that this is mostly going to talk about AI-designed drug discovery and big data and other such topics. It will get there. But there are many more such opportunities here. So we're going to start with some ideas that I think are direly under-discussed, underutilized, and theoretically already possible. The first is in literature searches. When I explain how fundamental scientific research works, the whole ecosystem of fundamental discovery to experiments to writing papers to publication, one question my dad has routinely asked is, okay, but who is sorting through and reading all the papers at the end? Who is the person collecting all the discoveries and working out what's important, what's not important, what ideas are worth pursuing? Who is the person who is putting ideas from different labs together, different countries together, and coming up with new directions? In some senses, it's everyone. We all read literature, present at conferences, hear ideas, and are inspired by these interactions. We write literature reviews on areas of our own expertise, combing through papers to come up with summaries and insights for everyone else. Pharmaceutical companies search through conferences and journals for promising new targets to follow up on. Journals and reviewers make decisions about which papers are most important and relevant, and those judged to be most impactful get sorted into the most viewed journals. In this sense, science works as a live information engine, a highly distributed machine working collectively across its constituent members. In another sense, though, it's also no one. No one has read all the papers. No one actually has the time to properly read much literature at all. Even if you read 100 to 200 papers a year, which would be truly an absurd amount, it would pale in comparison to the thousands of articles published on dementia research every week. Sure, not all of it is relevant, but even small subfields still have insurmountable and growing avalanches of information that no human can consume, interpret, and integrate. Furthermore, if you limit yourself to reading only those articles relevant to your own work, you miss all the ideas from adjacent fields, or even completely different fields that may nevertheless apply and give you a breakthrough. Reading articles or going to talks at random is just that, random. Not particularly efficient and not particularly effective. Putting together literature reviews, especially meta-analyses and systematic reviews, takes a huge amount of time and effort, and even when they're non-exhaustive. Moreover, many of us are limited to reading literature in one or a couple of languages, but there is so much more out there. There is simply too much information to sift through, and not enough time nor physical capacity to keep up with or process it all, let alone find all the things that are even relevant to you. Here is where AI comes in. We should be able to have AI language models trained and constantly updated on the entire of the dementia research literature, and neuroscientific and broader scientific literature for that matter. These should be able to answer questions about the directions the field is heading in, summarize latest findings, and find patterns in the data. This is just the basics. AI models should be able to suggest the best and most relevant papers that might have ideas, data, or methods that I'm interested in. AI should be able to read all my papers and ideas, and then tell me who in the world is doing similar research to me, who is doing complementary work, and who we best to collaborate with. and who is potentially already about to publish the work I'm going to start on soon. If I'm stuck on a research question, an AI that's read all the scientific literature ever produced should be able to tell me who would be best placed to answer it, even and especially if they're not from the dementia research field themselves. It doesn't have to be big picture theoretical questions either. Imagine I want to use a commonly used drug like rapamycin in my cell model to test the function of a process like autophagy, but I don't know what concentration I should use. AI, having read all the papers, should be able to give me a list of all the studies that have used rapamycin in cell models, what concentration they used, what cell models they used, and what the effect was. Same if I want to try using a new antibody I haven't used before. My life would be so much easier if I could just ask an AI to list all the times a particular antibody was used, what concentration it was used at, what kind of sample it was in, and show me what the images looked like so I could assess for myself what I should do. But what if I'm interested in a particular topic or concept? For me personally, it might be selective vulnerability, or it might be differential vulnerability, or maybe selective neuronal vulnerability, or regional vulnerability, or whatever keywords the authors chose to use. Should I search for induced pluripotent stem cells, iPSCs, iPS cells, HIPSCs? How about if I want to search for papers on autophagy, or macroautophagy, or lysosomes, lysosome, endolysosome, lysosomal function, endolysosomal network? Can you tell this has frustrated me before? The point here is that trying to search for terms without standardized terminology is a pain in the neck. But AI-powered natural language models should be perfectly able to interpret what I'm interested in based on my question, but also what it knows about my research and interests and research history generally, and then go and find everything relevant to that, regardless of the specific terminology or exact keywords used. What about when we do a literature search for papers on a particular topic, and the search returns 15, 30 pages of results? Are you going to read every title, every abstract? Will you click all the way to page 15, page 30? What if the answer you were looking for was on a paper in page 11, which you'd glazed over while you scrolled through? So much literature that may be useful has likely been missed or overlooked over the years. Similarly, it may be easy to think that something is a novel idea worth pursuing, when in fact it was examined before, and you just didn't come across that particular paper. Worse, it may be already under investigation by a different lab, and you didn't happen to be at a particular conference session when they presented the progress they've made so far. We could have AI that has read all literature, all conference abstracts, all departmental profiles, all lab websites, project listings, grant approvals, and so on, and these could help on this front. AI has issues that need to be recognized here when it comes to hallucinations, i.e. spontaneously coming up with information that has no basis in fact, but sounds plausible. This issue does somewhat undercut the potential use for AI in summarizing information from papers or producing literature reviews from scratch. However, designers of these large language models are already finding ways to mitigate these. Getting AI models to provide links to sources or search for exact quotes is one way of reducing this issue, for example. This also makes them readily fact-checkable. Moreover, as AI models get better and are trained on more data, error rates have been decreasing, with GPT-4 producing a 3 to 5% error rate and models improving on this already. The use of AI here isn't to provide a perfectly accurate summary of content, though. That is one potential application, but really what we wanted to do here is to prioritize, filter, and sort through the vast world of scientific content for us, providing starting points for us to spring off and investigate ourselves. To be the one person, as my dad might imagine it, who has read and interpreted all the literature, has made all the connections, and can point them out to people as needed or relevant. Number 2, enhanced conferences and collaboration. As alluded to earlier, another key application of an AI language model that has read all of the literature should be to circumvent the randomness of collaborations and conference meetups. We've all heard the idea that when you go to conferences, sure, you're ostensibly there for the talks and posters, but really it's the water cooler chats, the people you bump into in the corridor outside. That's where the real action happens, where collaborations spark and chance meetings burgeon into new discoveries. This view of conferences is absurdly romanticized and inefficient. If I'm going to a conference, I want an AI model to have read my abstract and have read my bio, as well as the abstracts and bios of everyone else there, and then suggest to me whom I should talk to. I want to be able to tell an AI chatbot what questions I'm interested in and have it tell me who is coming to this conference that can help me. Maybe these things are easier at small meetings, but especially for big conferences with thousands of people like ADPD, FENS, AAIC, SFN, and so on, this will be invaluable. I cannot possibly read through all 1,500 abstracts for ADPD, and even searching through all my key terms for the abstracts is a day's worth of work, let alone all the talks. Surely an AI that understands what I'm interested in can suggest what talks I might want to go to, and then, having looked at both our conference programs, suggest times to go and meet people that might be good to collaborate with when we're both free. If we want, AI should be able to point out to us who at a conference we've talked to before at previous meetings. It could be able to know which papers I've read and point out authors in attendance whom I might want to talk to. It could even be aware of whom I follow on social media, whose posts I interact with, whose work I admire, and whom I want to meet in person, and probably to reach out to them when we're attending the same meeting. Networking help. And yes, these ideas are inspired by all the times I've been at conferences and realized only too late that the person speaking wrote one of my favorite papers, or the times I've glanced at someone's name tag as they walk past, only to realize that they are someone I'm not going to talk to. All the times I've missed or overlooked a talk by someone that in retrospect would have actually been really interesting. But it's also inspired by all the times I've found myself in a random talk or in front of a random poster, not expecting it to be particularly interesting or relevant, only to find it absolutely fascinating and an important piece to my project, or using a method I could apply to my own experiments. We shouldn't be relying on random chance for these things to happen. Excitingly, conferences like BNA International Festival have already begun trying AI-based recommendations of people to contact and sessions to attend based on keywords and submitted abstracts. While not great in its first iteration, with limited data to work with, the premise has huge potential. To be clear, the idea here is not simply an AI gimmick for conferences. The hope is that AI can help better connect scientists from all around the world and enable us to stop relying on random chance and produce more fruitful collaborations. The current scientific paradigm has scientists in an uneasy middle ground between wanting to reach out and collaborate on the one hand and competing to publish new discoveries first, holding methods and data secret until then on the other hand. AI could change all of this, with an ability to predict who is best suited to working on particular questions, or who may already be working on or about to publish certain projects, based on their previous papers, funding applications, hiring actions, conference presentations, equipment purchases, and existing collaborations. This could also make finding new labs and projects vastly easier for students and postdocs in search of projects and jobs, or a home for their ideas. Instead of trawling through department faculty profiles, relying on word of mouth, or hoping to randomly bump into someone at a conference, AI should be able to tell us, based on a brief project outline, whose lab has the funding, the equipment, and the expertise necessary to house such an idea, and who else might be able to collaborate. If a lab is looking to hire someone with a particular skill set, AI should be able to tell them who at a conference would be a good fit and should be good to talk to. The potential for AI to transform science from a highly competitive landscape where information is hoarded and multiple scientists race in parallel on similar ideas, to a more collaborative one where scientists all over the world become connected in a more targeted and transparent manner is huge. Moreover, the potential for AI to transform collaboration and discovery from a semi-random exercise that largely benefits those with the funding and availability to go to conferences and proactively reach out to people, to a more targeted and equitable system is also great, and I'm excited for the benefits this will bring to research. Number 3, analysis of big data. The above applications of AI are more on the meta-science front, or how the process of research itself works and how it might be improved. But what about the experimentation and data analysis itself? Here too, AI is already becoming increasingly important and useful. Datasets are ballooning. Studies now involve more patients, more cells, more genes, more proteins, more longitudinal time points, more electrodes, more images in high-throughput screens, and high-density readings from wearable devices. This is largely a good thing, I think. The brain is the most complex thing in the known universe, and these are among the most complicated diseases confronted by modern science. We need high levels of granular data across large populations to inform our understanding of what are highly heterogeneous conditions. Using AI to analyze these datasets isn't just useful, it's necessary. AI can also not just integrate individual datasets, but also find subtle patterns across multiple modalities. Consider the case of mixed pathologies, as described in a previous blog. There are many dementia syndromes, driven by many different diseases, featuring many different combinations of many different pathological changes. These pathologies, be they protein accumulations, vascular lesions, metal iron deposits, synaptic loss, inflammation, glial scarring, or neurodegeneration, may manifest in different regions of the brain or in many different cell types, and this all likely informs the clinical presentation and progression of the disease, as well as a patient's response to different therapeutics. These are all likely driven by a patient's genetics, their lifetime exposures, e.g. to stress, air pollution, infections, and other toxins, and their behaviours, such as diet, exercise, sleep, education, sociality, and so on, and the biophysics of the pathologies in question. There are patterns to be found amid all these data points, and a huge opportunity to have a more personalised understanding of what causes dementia in different people, though not one that humans can achieve alone, without AI to sort through all this data and find the patterns. AI excels at quickly finding subtle changes and patterns in large volumes of data that we might not even think to look for. Integrating AI into analysis has enabled us to vastly increase the power of our studies. Compare the paradigm of neuropathological examinations in the past, where a vast team of researchers would have had to manually count stained cells and physically measure neurites if you want to measure patterns of neurodegeneration in the brain. This is both enormously time-consuming and expensive. Now, this process of image analysis that would have taken multiple people days or weeks to do can be done by one person and an AI in minutes, enabling vastly more samples to be processed in far shorter a time. Some might be concerned about AI automation replacing the jobs of scientists, but I would argue that there are much better things for researchers to be doing than counting cells across hundreds of pictures. Moreover, AI-driven automated image analysis can examine not just number of cells and neurite length, but all kinds of image features, from change in neurite branching and thickness to other architectural changes we might not have considered, identifying changes and patterns that would have been missed otherwise. Applications like cell painting, where various organelles and other structures of cells are all stained in different colors, allowing AI algorithms to examine hundreds of features of the cell at once and look at things in combination. Identifying differences under various conditions are enormously powerful for taking an unbiased approach to understanding the complex fundamental biology of these diseases. Consider also techniques involved in the recording of live brain cell activity. Understanding neuronal firing patterns is crucial to the fundamental functioning of the brain, and is one of the changes that is first thought to occur in dementia. Previously, this might have involved sticking a needle into individual cells and performing slow, painstaking recordings in low-powered experiments, measuring only tens of the possible millions of cells. Alternatively, it may have involved recording from scalp electrodes on a patient, reading out brainwaves using EEG, and hoping to see dramatic changes. Now, using technologies like multi-electrode arrays, neuropixel probes, electrocorticograms that record from thousands of tiny electrodes inside the skull, and EEG arrays with as many as 256 electrodes sitting outside the head, we can collect enormous amounts of data about the functioning of many cells at the same time. This kind of signaling information, however, is unfathomably complex. The data would be largely useless without AI to analyze it. Maybe we could identify big trends by eye, but could you really be confident that you can find all the patterns amid 256 separate electrodes recording brain activity? Could you really tell the difference between the firing patterns of a collection of hundreds of cells firing in a disease versus a healthy state? AI enables us to find the earliest, the most subtle, and the most obscure changes that may pertain to disease. It's capable of sifting for patterns in firing rates, amplitude changes, interspike intervals, synchronicity, changes in oscillation frequency and power spectra, and all kinds of other features that are beyond human capacity. Another interesting example is in speech changes. There's increasing evidence that speech can be a highly sensitive biomarker for a myriad of diseases and other health states, from colds and flus to mental health conditions, pregnancy, and of course dementia or movement disorders, not just aphasias. Changes could be in the sound of speech, the speed and cadence and rhythm with which a person talks, their pronunciation of certain words or sounds, and even changes in sentence length, vocabulary, and fluency. This is a perfect use case for AI, interpreting different modalities of data like sound and natural language processing, and analysing huge amounts of longitudinal data and searching for subtle patterns and changes occurring over years that may be missed and not particularly obvious to a clinician in a 5-minute observation. AI would then be able to tie back different patterns of changes to different diseases, enabling us to develop new, non-invasive, passive, sensitive, and potentially highly specific biomarkers for disease. To be explicit, analysing this kind and scale of data, be it genetic, structural, electrical, sonic, linguistic, or anything else, would be impossible without AI. These algorithms enable a huge scope of new research, better powered at large scale for lower cost, which could greatly increase our understanding of complex disease biology and the speed at which we do so. Part 4. AI simulation and modelling. Separate to the analysis of large data sets is what I have broadly categorized as simulation and modelling. This goes one step further from pattern detection amid large data sets and starts to make predictions, another strength of AI. This is all manner of applications, the most obvious of which might be AlphaFold. This AI program, built by Google's DeepMind team, is trained on all the known protein sequences and structures to predict structures of proteins we don't yet know the structure of, and to predict how protein structures might change given certain mutations or conditions. Experimentally determining protein structure is extremely difficult and time-consuming. AlphaFold, in predicting the structure of all known proteins, tens of thousands of different proteins, has completely changed the field. Understanding the three-dimensional structures of proteins is critical for deciphering their functions, interactions, and role in disease pathology, especially given that the hallmark of neurodegenerative diseases is the misfolding and aggregation of various proteins. Moreover, understanding the structure of proteins, not just those directly affected by disease, but also those involved in disease-related pathways, enables us, and by us I mean AI, to identify target sites for drugs, antibodies, and other means of intervention. AI simulation and modelling techniques can accelerate the drug discovery process by predicting the binding affinity and efficacy of simulated drug candidates targeting specific proteins involved in dementia. Using computational methods, such as molecular dynamics simulations and virtual screening, researchers can identify promising drug candidates, optimize their chemical structures, and predict their pharmacokinetic properties, all before conducting costly and time-consuming experimental studies. This takes us to the potential for so-called in silico experiments. Rather than having to house animals and going through the ethics concerns of conducting experiments in vivo, and rather than having to go to the expense and effort of painstakingly growing cells for months to do experiments in vitro, some experiments can be simulated on a computer in silico. This is still in relatively early days of development, but is already being applied to drug screening applications where experiments can be conducted at vast scales, much more quickly and cheaply, to identify top candidates and then validate them in real-world experiments. In the future, these AI-powered simulations may allow us to test hypotheses, simulate different biological processes, test the effects of genetic variations or environmental factors on cell function and disease progression, reduce our use of animal models, and more. Conclusions. This series was, in part, inspired by the AI UK conference debate on whether AI could improve dementia research and patient outcomes. While the debate itself largely focused on potential patient-facing applications in dementia diagnosis, as discussed in a separate blog, the reality is that many applications of AI are not in the future, but are already in use, and already benefiting dementia research. AI-driven analysis is already transforming how we understand disease and enabling us to integrate larger, more complex, and better powered datasets in ways that would have been previously impossible. Its ability to predict and simulate based off vast reams of collected data is already being put to enormous effect, with huge potential for accelerating target discovery through more efficient and expansive literature searching ability, huge potential for drug discovery through molecular and target simulations, and huge potential to make the whole process much faster and cheaper through using in silico screening and filtering. This also has not been an exhaustive discussion. For example, AI could also help dementia research by spotting scientific fraud more quickly. Data sleuths are great, but there are not enough of them to be able to cover all research at pace, and the field has already been marred by major fraud scandals. AI automation of basic research admin tasks could also free up time to do more actual science, which would not only be beneficial for the pace of science itself, but also the rate of people dropping out of research in frustration at the admin burden. These uses are not without their risks and concerns. AI itself remains expensive both financially and environmentally, though less and less so. And these, as well as privacy concerns, I've discussed elsewhere. Pitfalls in hallucinations should be taken seriously, though these are already able to be overcome if AI is used to generate suggestions rather than summaries, and thus can be easily fact-checked. And everything will eventually need to be experimentally validated anyway. Outside of the biology itself, the potential for AI to transform how the process of science communication, and science collaborations work for the better is also immense. These, in particular, present minimal risk. Most of the data these models will be trained on are public, so they don't have the same privacy concerns that patient-facing applications do, nor the same risks of harm. Ultimately, this is largely why I actually believe that AI can be a game changer for dementia research, and the benefits outweigh the risks. Neurodegenerative diseases driving dementia are simply too complicated for our simpler tools. Patient conditions are too heterogeneous, and mispathologies and the multifactorial etiologies too varied for us to take anything less than a big data approach. This requires AI, and in doing so, could allow us to personalize dementia care, identify disease far earlier than we currently can, and study the earliest changes of dysfunction that are presently inaccessible to us. These things might be possible without AI. It would take far longer, cost far more, and require a vastly bigger scientific workforce than we currently have. Even if we decide that AI is too risky to be applied in patient-facing settings, such as diagnosis and clinical decision-making, the potential of speeding up drug discovery and accelerating fundamental research through targeted collaborations and more efficient, comprehensive literature searches is immense. It would transform our engine of knowledge from one running on chance discovery to one designed to best harness and coordinate the collective intelligence of the scientific community. It represents a paradigm shift in how we generate scientific understanding of the complex fundamental biology of disease that can accelerate our journey to discoveries, ultimately improving outcomes for researchers and patients alike.
Thank you for listening. Join the Dementia Research bloggers and share your own views.